Um, Welcome everyone. Thanks for joining us today for our live webinar, which is going to be presented by Ruth Andrew from University of Edinburgh, called Visualising Steroids, Images of Endocrinology. My name is Val O'Donnell. I'm based in Cardiff in the UK. Webinar is being recorded as usual, and it'll be uploaded onto our website and YouTube channel within the next 48 hours. And as usual, it'll be around 48 minutes with about 10 minutes at the end for questions. So today's webinar is being sponsored by Cayman Chemical. We're grateful for their ongoing support, and I'd now like to introduce our speaker. So Ruth Andrew holds a chair in pharmaceutical endocrinology at the University of Edinburgh, and she's director of the Clinical Research Facility Mass Spectrometry Core. After qualifying as a pharmacist, she studied for a PhD in pharmaceutical analysis using GCMS to profile catecholamines in hypertension. In '94, she joined the endocrinology unit at the University of Edinburgh to develop further interests in mass spec and develop its use in steroid profiling in cardiovascular disease. And that has been an enormous amount of work for which Ruth has done a huge amount of work in this area that is now, I think, leading towards clinical applications, which maybe we're going to hear some of today. So really looking forward to that. Since then, Ruth investigated regulation of glucocorticoid metabolism and has focused on the role of hepatic 5-alpha reductase in diabetes, also developing dynamic methods to quantify these metabolic pathways using stable isotope tracers. And I was looking at some of your papers earlier. Is that it maybe or may not be the PET scanning work that you did recently as well?
Oh, yes.
A combination of mass spec and PET scanning. So not just doing mass spec, but also doing PET scanning and MRI as well. So a fantastic array of methods for profiling these molecules. Ruth takes an active role in teaching both honours students and postgraduate students and contributes to university governance. She's the General Secretary of the Society for Endocrinology and acts on grant panels for the Chief Scientific Office in Scotland, FWO, Flanders, NCN, Poland, and the Commonwealth Commission. She's a review editor for the British Journal of Pharmacology and a specialty chief editor for Systems Endocrinology. So thank you very much, Ruth, for agreeing to present today. I'm very much looking forward to your talk. I guess we'll be looking at some really cool images of, yes, I love imaging, so I'm looking forward to seeing that. So images of endocrinology, over to you, Ruth.
Thank you very much, Val, for that kind introduction, and everyone for coming today. So yes, I think Val's covered my general areas, and what I was going to do today is tell you a little bit about how I've investigated steroid biochemistry at the outset, just to talk about the preclinical and translational applications in that field. And then I'm going to move on to how we've tried to tackle measuring steroids at a tissue level using mass spectrometry imaging, and finish up with a broader vision for lipidomics in that space. So that's a quick tour of what's going to happen. So my career has been based around glucocorticoid hormones, and this is the main one in humans, which is cortisol. So this is one of very many steroid hormones that we have. They form a subclass of lipids, and they have got particular roles in health and disease. And this is what you would regard as their healthy responses are really important for immune responses, stress response and development. I've highlighted metabolism because I'm going to be talking about that in more detail today. So we need them for health, but they can go wrong whether they're in high or low levels. And we've particularly been interested in what happens when your glucocorticoid levels go up, what goes wrong. And in particular, what we find is people are prone to having cardiovascular disease and diabetes, as well as problems with immune suppression and cognitive diseases. During pregnancy, high glucocorticoids can cause problems with long-term programming of diseases at later stage in life. So we know this because of various more relatively rare endocrine disorders where some people have tumours that produce too much cortisol or are driven to produce too much cortisol. And this leads to a spectrum of these features. And in particular, it leads to a condition that looks very much like the cardiometabolic syndrome. So our constellation of type 2 diabetes, obesity, hyperlipidemia, and hypertension. So this is reversible in these endocrine patients. So that's good. We know we can reverse these changes. You take away the tumour. We see this also in people that take glucocorticoids to treat asthma and high dose. They get similar problems. Again, it'll stop when the drug treatment stops. So this has led to hypotheses that perhaps people in the general population who are prone to developing these types of disorders might in some ways be exposed to glucocorticoid excess. And that has underpinned a lot of the work I've done and trying to unpick this conundrum using mass spectrometry. So first of all, we need to understand what's a healthy way that glucocorticoids are produced, and that's regulated by the hypothalamic pituitary adrenal axis. It's an endocrine axis driven by the brain, and the steroids are produced by the adrenal gland. So this allows us to control our daily rhythms, our responses to stress, and it's a tightly controlled negative feedback loop. Interfering with that axis for therapy is a bit of a blunt tool because, for example, if you suddenly get exposed to a stress, you then are less able to respond to it. So it's not a particularly good approach. But there are other ways to manipulate the levels of glucocorticoids, and that's by looking at the fine-tuning mechanisms that we have in tissues. Now, I'm going to talk today about enzymes carrying out this fine-tuning, but there are alternative ways. For example, there are proteins called binding globulins that deliver the steroids. There are pumps that pump the steroids in and outside cells. But I'm going to talk about the enzymes that alter the steroids between their active and inactive forms. So this little cycle goes on in tissues, and we can interfere with these enzymes to either suppress or raise the glucocorticoids at local levels. Now, these enzymes occur in a number of different tissues, but in particular interest to me, they occur in metabolic tissues like the liver, the adipose tissue, the skeletal muscle, the brain, and the kidney. So all these tissues, you've got this fine-tuning possibility. So when I started my career, one of the enzymes that was very well known in this field was 11-beta-hydroxysteroid dehydrogenase type 2, and it was discovered in the kidney. And it takes cortisol, our active hormone, and then converts it at the 11 position. Just see if I can get the pointer actually. At the 11 position from the hydroxy to the ketone. And accordingly, you inactivate the hormone. So that's by far from the only enzyme, though, that actually carries out this type of conversion. And I became interested in another set of enzymes that actually act on the A ring of the steroid, which is this ring here. And it can convert this double bond into a tetrahydral form, which is the double bond and the oxygen reduced, either in the beta or the alpha orientation, which I'll come to in just a minute. Now, we became interested in that when we were actually profiling this original enzyme by GCMS, which was my first role in Edinburgh. And here's one of my original GCMS profiles, where we can unpick these two metabolites within the urinary profile, pick them up in urine, and you get different patterns. So, for example, what we tended to find that people that were lean had more of the 5-beta form, and people that were obese had more of the 5-alpha. So it's typical, sort of like as you do with pediatric profiling of steroids, you can see the pattern change. We didn't really know at the outset if this was really important or not, but we studied this in a number of populations, and we consistently got this pattern. So again, in the 90s, we quite often presented this ratio of steroids. This was sort of around the sort of John Honor, Cedric Shackleton papers. There's a lot of this type of analysis where we look at the 5-beta and 5-alpha ratio. This was the way it was presented. And we found in people with an increasing abdominal circumference, these ratios fell both in men and in women. And that was primarily driven by an increased amount of these 5-alpha reduced steroids. So say we didn't know if that was important or not, but it was something worth following up. And it started a series of experiments for me trying to unpick what was the role of the enzyme that formed these 5-alpha steroids in metabolic disease. So we just take a little bit look at the structures. What we're doing is we can convert cortisol through to its 5-alpha form at the 5-alpha position here. And also we can then carry on with 3-alpha HSDs to form the hydroxyl at this point. So the actual intermediate here is only very transient, usually found in this tetrahydral form. These are quite similar enzymes to that we find in bile acid analysis. For example, although it's more the 5-beta form, you tend to find it in that setting. So that's the reaction, and this is what the actual steroids look like. We've got different stereochemistry orientation at the 5 position, and hopefully, yeah, so it works. What you see is they actually have quite different shapes. So the 5-alpha steroids is quite a flat steroid. This has got a very similar orientation and shape to the original parent glucocorticoid, whereas a 5-beta steroid has got a really skewed shape. And you could imagine if these two form and they interact with proteins in the body, they'll interact with quite different subsets of proteins. So the 5-beta steroids have had some exposure in terms of binding to receptors such as like PXR, FXR, which actually what we find is the 5-alpha steroids are much more likely to bind to the same receptor as their original steroid. So in this case, the glucocorticoid receptor. So we were interested by this, and we asked ourselves, well, what's the purpose of forming more of these 5-alpha steroids in obesity and the metabolic syndrome? We knew they formed. Was it that we were forming an active metabolite that was causing the condition to get worse? Or was it the body trying to get rid of what was known as the active hormone cortisol as a protective mechanism? And the way we chose to investigate that was simply by inhibiting 5-alpha reductases and studying what happened. So we were fortunate in this case because there were drugs available that were already clinically being used to inhibit these enzymes, and this was not in the setting of glucocorticoids, but in the setting of androgens. So before I skip back a step, this was our glucocorticoids getting converted through to the 5-alpha glucocorticoid here. We did spend quite a considerable amount of time, still do, looking at the activity of this 5-alpha glucocorticoid, and we find that it is not a metabolic glucocorticoid, so it's not causing metabolic damage, but it still retains the anti-inflammatory properties of the parent steroid. So this has led us into some studies of what are known as selective glucocorticoid receptor modulators, which are molecules that interact with the glucocorticoid receptor by triggering different combinations of pro-regulators, and thus only subsets of the mechanism of action of the main parent glucocorticoid. It's similar to SERMs that are used to manipulate estrogens selectively in breast cancer. So it seemed that our metabolite wasn't metabolically active, so it didn't seem likely that it was the problem. It's more likely that if you inhibit that what we were doing was trying to get rid of the active hormone, which was the metabolically active compound. So just to return to what I was saying about alternative mechanisms, we had drugs because the 5-alpha reductase also metabolizes a number of other steroids, and I'm showing you here the A-ring, which is similar in testosterone and the metabolite 5-alpha dihydrotestosterone. Here's the whole steroid. This enzymes also metabolize progesterones, mineralocorticoids, and a number of other hormones as well. But I'm just going to tell you about androgens. So the drugs were developed to actually inhibit this conversion of testosterone to dihydrotestosterone. Dihydrotestosterone is a much more potent androgen than testosterone, and where it forms in the body, it drives androgen action. And that's important in the prostate, where the prostate is an androgen-sensitive gland. It swells and grows in response to androgens. And this is a problem in older life. It's a very prominent problem. For example, you see that 80% men over 80 will have this swollen prostate. It causes pain and urinary complications, and one way to treat it is to stop the prostate swelling, and you could do that by blocking the action of androgens. So drugs were developed to block 5-alpha reductase specifically to try and reduce the swelling of the prostate. They're also used for hirsutism in women and in some cases of prostate cancer as well. So it was quite fortunate for us because we had clinical tools where we could actually look in individuals and actually study whether or not these drugs, and by inhibiting the enzyme, were altering metabolic disease. And nobody had looked at these people that were taking these drugs to see whether they had any change in their metabolism. So we went through a series of translational studies where we inhibited the 5-alpha reductase enzymes in order to see what happened to metabolism, proposing that we were going to get a build-up of this metabolically active cortisol. Now, so we studied this first in animal models. We were lucky that there was a 5-alpha reductase knockout mouse formed by Mala Mahendra in Dallas. There are 2 isozymes, in fact there's 3, but there were 2 at the time that were known, and she had knockouts of both. We chose to study the type 1 knockout because that was the enzyme that was present in all the metabolic tissues. And in blue here, you can see that the enzyme, the wild type and knockout mice, normal diet, actually have a very similar sort of weight trajectory over a period of 6 months. You put them in red, the wild types on a high-fat diet, what you find is obviously they gain a bit more weight. But if you put the knockout on a high-fat diet, we found it gained considerably more weight. So that fitted with our hypothesis that we were failing to inactivate glucocorticoids in this setting. You can see it by DEXA scans and just visually, to be honest. It took a while. It wasn't a quick obesity, but it took a while, and it generated over about 6 months. Now, we have also, this is male mice, we've also studied female mice, and if anything, they're more susceptible. They do gain weight, and they don't need the high-fat diet to cause the change. So there is a subtle sexual dimorphism there, but the same phenotype does develop. Now, we studied these mice by infusing glucocorticoids to see whether or not the animals accumulated the glucocorticoids at sites where there could be metabolic complications. And you can see here as measuring glucocorticoids in the liver and adipose, where in the stripy bars, you can see the knockout at higher levels. Now, this was probably one of our earliest attempts at trying to measure tissue steroids, and it really was very challenging. Steroids don't ionize very well by mass spec, and it's actually quite difficult to pick them up in complicated matrices unless you do thorough cleanups. So particularly with adipose, removing the lipids to avoid the matrix effects was quite difficult. So that we did it, but it wasn't an easy thing at the time. This was probably a good 15 years ago. So we've studied them in more detail by metabolically phenotyping, for example, insulin responses to glucose tolerance tests. So here you give a glucose load and see how the insulin responds. If you're insulin sensitive and healthy, you don't need that much insulin to respond. Whereas if you're a bit insulin resistant and heading towards diabetes, you need more insulin to respond. So there wasn't any difference in a normal diet in blue here between our wild type and our striped knockouts. Oops, sorry, going backwards. But in the actual high-fat diet in red, you can see that the knockouts started to need a greater insulin response to control their glucose. They had normal fasting glucose. They weren't actually diabetic, and then we've never seen them develop diabetes. Actually, they have never gone on to that. But they did need a much higher fasting insulin to control their glucose. So they had lots of features of an animal prone to diabetes, type 2 diabetes. You could also see that in the liver, where you see the hepatic manifestation of diabetes, where we collect triglycerides, and that's a pathway for the liver to gradually become inflamed and potentially cancerous in the long run. We tested the inflammation over here on the right using a carbon tetrachloride model, where you inject that and you can see whether or not there's a fibrotic response. And you can see in the knockouts here, there's a much greater fibrotic response. So this could be to do with the lack of that inflammatory steroid, but it's certainly the knockout was much more prone to liver dysfunction associated with metabolic disease. It isn't the case for cancer, but it is for the fibrosis. And another group in Birmingham at the time also showed similar things. So on that setting, we felt we had a situation where it appeared that this enzyme was regulating the levels of active glucocorticoid. Inhibiting it made you more prone to metabolic disease in the rodent model, and that was therefore interesting to follow up in the human. Now, in the human, we were lucky we had the drugs that we could manipulate the enzymes. And I just want to introduce here the two isozymes. These are the ones that are best recognized. There is a third, but these are the ones that are best researched. So you can see that in the prostate, the 5-alpha-R2 is the most dominant isozyme, and so therefore the first drug that was developed actually just inhibited the type 2 enzyme. There's not much type 2 enzyme in human metabolic tissues. There's a bit in liver and a bit in muscle. So actually the type 1 enzyme is more abundant, but there's another drug that got developed called dutasteride that inhibited the two enzymes and thus helped the prostate disease better, but it allowed us to have an enzyme that we could knock out the principal isozyme in liver, adipose, and skeletal muscle. So we proposed to check whether or not patients taking these drugs were becoming insulin resistant. And we did that initially in an experimental model just over 3 months in healthy people to see how they fared. And we compared both drugs to another drug called tamsulosin, which is used to treat prostate disease, but it's nothing to do with 5-alpha reductase. So we tested it whether our dual inhibitors could influence insulin resistance in men. It was aged quite a wide age range for 6 men. And we made baseline measurements by a number of different detailed metabolic phenotypes. Give them the different inhibitors, and then measured them again 3 months later. And we had a relatively healthy group to start with. There were BMI about 25. There were no diabetic people within it, and perfectly normal blood biochemistry. And what we then did was study them over the 3 months. And these graphs here are all of this style, so that shows you the data before the 3 months, to after the 3 months. So it's a difference. And if the data deflect upwards, that means they got worse. So in this case, the group you could see in pink gained body fat. So that was our dutasteride group where we had the dual inhibition of both enzymes. You didn't see so much with the other, with just the finasteride, which is indicated in 2. That's the type 2 inhibitor, or the control C. And the same thing happened with other metabolic parameters as well. So we saw change in fasting insulin that got worse in dutasteride, and C-peptide and HOMA-IR are longer-term indices of insulin resistance that also got worse when you gave the dutasteride. So that matched with our animal model, that we showed that this 5-alpha, probably 5-alpha type 1, was particularly regulating the glucocorticoid levels, and lacking that enzyme or inhibiting that enzyme made you prone to metabolic dysfunction. Now, our lab does a lot of dynamic stable isotope flux measures, as Val said, and one of the things we support are euglycemic hyperinsulinemic plants with stable isotope tracers. And what this model does is it allows you to infuse insulin at different levels and look to see how much glucose you dispose of. So if somebody's very insulin sensitive, at a certain level of insulin, you'll dispose of a lot of glucose. If you're insulin resistant, you won't dispose as much glucose for that level of insulin. So you're basically looking at the interaction between the two, and you get the best measures of that if you add in stable isotope tracers of glucose into the mix. So we infused these different levels of insulin. You can see the rising levels here, and we managed to control the glucose concentrations, which is what we were aiming for. And what we needed to measure, therefore, was how much glucose we were disposing for a particular insulin concentration. So the experiment went well. We infused our tracers, which are D2 glucose and D5 glycerol, which allowed us to measure actual rates of disposal of glucose. And what this allows you to do is look at the disposal of glucose in the peripheral muscle when you're up at this high dose of insulin up here. In the middle bit, you can look at lipolysis with your glycerol and hepatic glucose production here at the low dose, again, of the insulin clamp. So again, I've got another graph here that's showing you difference from baseline. And if nothing had happened to our patients, our data would track right along this 0 point. And what we're looking for is looking at our glucose disposal, and what that would make it better, and that would make it worse. So it's the opposite way around this time. And what we found at low dose insulin, we didn't really see much difference between our patients, but we found that actually our control group and our finasteride group got a bit better, whereas our dutasteride group got worse. So they were less able to dispose glucose when you're receiving dutasteride. It's quite interesting to see the control groups getting better, but that's a relatively common occurrence in metabolic studies, because quite often people start taking a little bit better care of themselves when they're getting scrutinized for their metabolism. So again, we found dutasteride was bad for us. That was and that suggests that men taking these drugs might be prone to developing more rapid insulin resistance and be on a track towards diabetes over a longer period. And these drugs are prescribed for 10, 15 years, so it's not a short term. This was only a 3-month intervention. So our next step was to look at a population level, where we studied men receiving these drugs for benign prostate disease over a period of roughly about 10 years was the first study we did. We studied a population database called the CPRD database, which is a prescribing database in the UK, identifying the men that were taking these drugs, the same groups again, the dutasteride, finasteride and tamsulosin, cleaned up all the data, adjusted for confounders like drug treatment, social deprivation scores. We made sure we propensity matched so that we tracked people with the same starting BMI, etc., same GP surgery, those types of things. And we studied how likely people were to get new incident diabetes over a period of, as it turned out to be, 10 years. And this shows you that we actually found an increased risk of diabetes. So this is our CPRD database. One would be no change in the risk of diabetes, which is what we see for tamsulosin up here. But actually both the 5-alpha reductase inhibitors caused a deflection to the left here of the risk of diabetes. It's roughly about a 30% change. So it's significant over a 10-year period that there's an increased risk of diabetes in this group. And of course, it's important to replicate. So we replicated in a group in Taiwan, which is interesting because it's got a different ethnic background again, adds weight to the biochemical finding. And again, we found this deflection that we've got an increased risk of type 2 diabetes in the people, new type 2 diabetes in people receiving the drugs. And you can see that here as a time course where both the finasteride and dutasteride incidence curves start to tail away from the tamsulosin and a slightly higher level of type 2 diabetes risk. It was interesting, we didn't see a difference between the two drugs in the longer term, but I don't think we really have a full explanation for that, except that there is the type 2 enzyme in the liver as well, but that seems to be consistent in our data. You can try to stratify that down a bit, and what we find is if we look at the patients depending on their BMI, the risk's really only present once you start to get overweight or obese. If you're actually healthy over that 10 years, you don't really see a significant increase. So it's quite important information for thinking about your patient cohort as they start receiving these drugs. Now we're continuing with these studies. This is as far as is published. The next slide is unpublished. I've got a new student currently looking to see whether when you've got diabetes, if that then predicts you having an altered cardiovascular disease or endpoints or death by myocardial infarction, stroke, etc. And we're also studying the rate at which diabetes progresses, like, for example, how long it takes for someone to progress from tablets to insulin. And I've not got all that data yet, but this is the latest hot off the press. And this is showing the risk of myocardial infarction. And you can see in blue with dutasteride that the myocardial infarction is a couple of percentages greater than the other groups. So there is an increased risk of some of the cardiovascular endpoints that go along with diabetes. This was done over a 15-year period. We're hoping now to look specifically in the diabetic cohort. This is a general population because it might well be that they are even more prone. So this has been quite an exciting project. It's been quite nice to take something right through the way through from animal research and I guess sort of hypothesis, something that we didn't expect to find, through to a finding that is actually impacting at the clinic. And what I've mentioned as I've gone along actually is the fact that we've been measuring steroid hormones all along the way. We're measuring glucocorticoids, checking androgen levels, and that in itself is really quite challenging. And I've got a graph here that just shows you on a log scale on the y-axis about the range of concentrations that steroid hormones exist. This is in blood. And you can see that our cortisols are sort of higher, but we've also got other steroids that are really low in concentration, like aldosterone or even estrogens, once you start inhibiting them in breast cancer in particular. So this is an extremely wide dynamic range of steroids in the profiles that we develop, and which brings challenges for instrumentation and particular challenges when you're working with very complicated matrices like tissues, because it's really quite hard to pick out those low abundant steroids against the tissue matrix. So although we regularly measure blood and urinary fluid analytes, one of the things we've tried to do along the way is find better ways to measure steroids actually at their site of action in tissues. And I'm just going to move on to show you some of the work we've done to try and develop better tissue measures of steroids. So we use a technique called mass spectrometry imaging to try and actually look at steroids in situ in tissues. And this involves using a laser, which we fire at tissues in a rastered pattern in order to collect the steroids in particular coordinates. It's not just steroids, but we're particularly focused on steroids. We make our analysis for this particular approach using Fourier transform ion cyclotron resonance mass spectrometry. See if I can get this to go. Yeah, and that works by making molecules spin in a trap, and smaller molecules spin slightly faster, and you can then use Fourier transform deconvolution to work out the exact mass. So this is quite important because this type of mass spectrometry analysis doesn't have any chromatography, so you're limited to just mass in trying to identify your molecules. So this is what the tissue surface looks like after we've done our analysis. You can see these ablated holes that the laser has made, and we can position the distance between these holes. We commonly work at 75 micron steps, but you can go smaller or higher depending on what you're doing. And within these, this now then allows us to collect the molecules in every single zone and recreate maps of molecules within whatever zone of histology we're interested in. So this is an example, one of our early examples, one of my students, David Kabichi, who looked at a drug that we were interested in because we thought it affected glucocorticoid action in brain. So we're working along with outside our drug development chemists here. And this is a mouse brain, sections across the mouse brain. Here's the control here. And then this is 1 hour, 4 hours, 6 hours after you've given the drugs. There's 3 separate mice. And you can see by the heat maps that this particular ion that we're picking up, which is the drug, is changing. It's changing in abundance, which you can relatively quantify. It's also you can also see exactly where in the brain it is. Now, at this point, this is quite early days. We're probably working with 150 microns. We do it slightly more detailed now. What's also important is these are extremely rich data sets. They don't just contain the one molecule that you're interested in. They contain very many other molecules. And so, for example, you can ask the data set, to look for metabolites that you might expect to find, hydroxylated metabolites. And we found, for example, these other metabolites according to cytochrome P450 localizations forming in the brain. So this is like autoradiography that the pharmaceutical companies do, but often the problem with that is the radio-labeled molecule you're looking at looks exactly the same as a radio-labeled metabolite. Here we can use the mass to completely distinguish them. And it's been a very powerful technique to help us localize molecules in tissues. Steroids, however, are always a problem because they don't ionize very well. And this was one of our very original attempts at doing the same thing with this is the rodent glucocorticoid, corticosteroid. You can see we've got a very, very poor signal here. So that's just simply because it's competing with matrix for ionization, and it's very hard to pick up. So we had to find new ways in order to enhance the signal. And both in this and our chromatographic approaches, we commonly use derivatization, which is a technique to add a charge tag or a tag that will charge onto the molecule so that you can see its signal more clearly in a complex situation. And this derives right back to some of the hydrozones, Girard hydrozones that were added on to testosterone by Shackleton, for example, for chromatographic purposes. So this type of approach can be adapted for tissue surfaces. So, for example, we've used these sort of hydrazone type molecules to look at our ketosteroids, where we can tag on to molecules like dihydrotestosterone onto this part of the molecule here. And for chromatography, it boosts the signal nicely. Dihydrotestosterone is a really difficult molecule to measure. It's in very low amounts, but we get really nice clear signals when we use this derivative. We get isomers as well, but we get nice clear signals. So we adapted that approach that we'd been using chromatographically to work on the tissues. And here's just the reaction that we use for the glucocorticoids. And that was really effective because here's the image I showed you before, and here's what happens afterwards. We get very nice clear signals, and we can see the signal strongly in the brain. So we've used this approach in very many different ways. I'm just going to do a little bit of a romp through a few of them. But what we have done is been able to pick up molecules that are really in very low abundance. So one of the molecules here, aldosterone, helps us control our blood pressure. It's really sort of very, very low picogram quantities, and so it's hard to detect by chromatographic means. So by derivatization, we are able to bring it to the fore. And one of the regions you might expect to see aldosterone, just by way of example, is the adrenal gland, where it's formed in the outer area of the adrenal gland called the zona glomerulosa. And here you can see it here around the outside of the adrenal gland, whereas if we look at glucocorticoids, they're appropriately positioned in the fasciculata or internally in the adrenal gland. So you can see these really low-level hormones because you put on this sort of shining tag to pick them up. DHT, I told you, was a difficult thing to pick up. And here's just an example of some testes images where we've managed to pick up testosterone and dihydrotestosterone. So that's our 5-alpha reductase reaction. And if you look closely, you'll see that these are actually in different parts of the testis section. One's in the tubules and one's in the interstitium. So this gives you a really fine detail as to where hormones are acting and are co-located or separated. I use this slide quite often just to highlight the importance of thinking about what histological features you need to pick up, because we had to drop to 50 micron imaging to see this. If you go back to 100 or 200, you can see how you lose that histological detail. This is a HCG stimulated testis on these two, by A and B, and that just won't give you the detail. And actually the seminiferous tubule is about 350 microns in a mouse. So you've got to think about what you do when you're doing this technique. It's not always necessary to drop to 10 microns, for example, but you need to just think, what are you interested in finding? But those testosterone images are really clear about what the value and the detail you can gain. So we've used this to study a number of different enzyme systems, and I'm just going to go back to this original cartoon I had where I showed you that, you know, glucocorticoids are activated and inactivated. I've already spoken about 5-alpha reductase, but I'm just going to tell you a little bit about these beta HSDs. There are two of them. One, type 2 inactivates glucocorticoids and type 1 activates glucocorticoids. So we focus on this type 1 initially. This is an enzyme that generates glucocorticoids in tissues, and it's been an important developing drug target, because if you can inhibit that, you can lower the levels of glucocorticoids in tissues in, for example, metabolic disease or cognitive dysfunction. And one of the areas that has gained a lot of traction in is there's a drug in clinical trials which is getting used for Alzheimer's disease, because it's supposed to be lowering glucocorticoid levels in areas of the brain, like the hippocampus, that are glucocorticoid sensitive for sort of neurotoxicity. So we were part of the team that developed some of the investigative brochure for that, because we helped show that the drug got to the brain through this mass spec imaging approach. So you start to pick up, this is the active inactive glucocorticoid, this is the enzyme, and here it's forming the active glucocorticoid over here. You can see that there. And then if you inhibit this enzyme, this was actually a genetic inhibition, you can see accumulation of that substrate. It's hard visually to see the difference in the actual active glucocorticoid, but the balance of the two gets altered, and that can be quantified relatively. And you can see in the turquoise that in our knockout mouse, we had about the ratio roughly halved. We could do that with the drug as well, and the dose we gave, we showed about a 20% reduction in this active to inactive balance. So that has fed through to improvements in behavioural aspects of Alzheimer's disease, and I'd say the drug's still going through clinical trials. In these studies also, we used some of our stable isotope tracers to try and find out which areas in the brain were more active in forming glucocorticoids. So we do this by looking at generation of a deuterium-labelled cortisol, and this is a section across the brain, and the regions lighting up in green are the areas that are more actively forming this steroid. So that's helping us understand which areas are at a local level generating their own glucocorticoid, and therefore might be more susceptible to drug treatment. And the hippocampus is one of the ones that's actually represented here. So I'll skip now to the other enzyme, 11-beta-HSD type 2, and just show you a little bit of imaging within the kidney. And this is an enzyme that inactivates glucocorticoids. So this is the sort of classic idea here. Glucocorticoid in the kidney gets converted to its inactive form, cortisone, by 11-beta-HSD type 2. And this is known to be a key process that helps us control our blood pressure, because if you get mutations in this enzyme, people end up very high blood pressure from birth, and polymorphisms are at play here as well. So the way this works actually is glucocorticoids, when they have two different receptors, at least, the glucocorticoid receptor and mineralocorticoid receptor. And mineralocorticoid receptor has got two ligands. Both cortisol and aldosterone can act on the mineralocorticoid receptor. And the sort of endocrine control mechanisms that generally control blood pressure and salt balance regulate aldosterone. So we really want aldosterone to be the key thing that's getting onto the mineralocorticoid receptor. So in tissues where cortisol and where aldosterone should control the mineralocorticoid receptor, there tends to be this type 2 enzyme to inactivate any cortisol that comes along from the blood, so that it doesn't inadvertently get onto the receptor and interfere with your blood pressure control. In cells with just GR, cortisol can act. So that's been understood through a series of sort of investigations of knockout mice and polymorphisms and mutations, but it's still not really quite clear in quite which cell types in the kidney are being controlled in this mechanism. And you can do so much by Western blots, etc. But what would be really nice is to measure the steroids in the actual subregions of the kidney, and that's what we set out to do. Now, originally we would have used our GCMS method with urine and just measured the metabolites of glucocorticoids, but that doesn't tell you anything about the renal structure or which types of cells are affected. And here's just a depiction of a sort of cartoon of the kidney, where you can see that really elegant structure. And all these tubules that zigzag back and forwards across the kidney with all these different functional zones, some of which have got the mineralocorticoid receptor. So one of my recent PhD students set out to apply mass spectrometry imaging to try and let us image where in the kidney we could find the active and inactive glucocorticoids. And here are typical images of the kidney that we get from mouse, where you can see that the active glucocorticoid sits in this sort of cortical, just at the inner cortex here, where it lights up in green, whereas the inactive one sits right in here in the inner medulla. So this inner medulla region is an area that's getting protected from glucocorticoids, whereas the pit on the outer side is where an area where perhaps glucocorticoids are interacting with the glucocorticoid receptor. Now, it is quite hard from those images just to see precisely which cell types are involved with this, and what we need to do is try to line up histology with regional function. Now, we can do it quite crudely by just drawing what the medulla is, the outer medulla, the inner medulla, and relatively quantify the steroids. This was an experiment where we manipulated salt, and you can see in low salt diet the glucocorticoids go up in the cortex, but that still doesn't quite tell you which tubule was involved, which part of the tubule. So we need to see that in more detail. And if you start to, this is work from Roger Brown, you can start to do that by co-registering with transporters and particular ion channels that only occur in certain cell types. So these are in situ hybridization that shows you potassium and sodium ion channels. And what we can try to do is overlay our steroid images against these different patterns of ion channels in order to identify which of them are seeing active and inactive glucocorticoid. Now, that is a major job of spatial clustering, which is still underway, but that is the ultimate goal with this work. So I think I'm still working on that, but that is where we're going. I just wanted to highlight again that these data sets are really rich. They're not just the steroids we're looking for. They're a whole lot of markers that are embedded in there as ions, that some of which we can identify and some of which we can't. But they come out with all these different patterns that tell you about the tubules. And, there's basically an omics data set that can be interrogated. So for particular, we have these omics data sets now for what happens in different salt diets. And we can look for markers of that won't necessarily be steroids, but they might be a very valuable biochemical information when you drill down to the detail. I'm going to finish off just showing you something with a slightly wider lipidomic profile, since not everyone's passionate as steroids as I am. And this is us trying to do mass spectrometry imaging just in this sort of broader lipid space. And so this is work of SPA, who's in picture up the top, who's working in the field of atherosclerosis. And what we're trying to do here is try to understand why some people that develop atherosclerotic plaques have got more vulnerable plaques than others. And by doing that, we're trying to look at the lipid composition of the plaque, but not just by taking the plaque out and homogenizing it up, looking at it in spatial detail. So atherosclerosis, very serious, very many deaths a year, and you build up the plaque, and some people, and at some point, the plaque might become unstable. So plaques are quite difficult to work with, particularly if they're calcified. They're really quite hard to cut. And we've had to work out ways, and we've had advice from Richard Goodwin's team on how to mount these plaques. We now use hydrogels. So we find this actually very helpful for a lot of tissues to get better sectioning. And that then allows us to mount the plaque in order that we can get decent sections. From that, we can do histology staining in a number of different ways, but for example, here's an H&E that shows you where the calcified region was, for example. We can drill down and image the calcium. We can look for where the lipid core is, where the smooth muscle, etc., and also our macrophages. And we can superimpose these images with our lipid imaging. So this is a lipid, for example, you can see in the interstitium, the intima, sorry. And whereas we can also do clustering so that we can work out which lipids, for example, are in the fibrotic cap and not in the intima. So this is, again, work underway, but we, again, are creating these really in-depth data sets for the different subregions in these clusters. We don't, as said earlier, we don't have chromatography, so we can assign putative identification based on mass. We do this, if we can, by FTMS, FTICR, followed by fragmentation. And then we also have ion mobility in our lab that we can employ to help us The lack of chromatography obviously is an issue in trying to resolve all the lipid species, but that is the workflow that we work with just now. But that then allows us to try and work out subsets of lipids that sit in plaques that are more vulnerable to rupture. So through a typical sort of PCA and volcano plot, we find that quite the molecules of particular interest are actually often sphingomyelins, ceramides, and some phosphatidylcholines. So we're looking at these in more detail, and we will be able to follow up by chromatographic methods if we need to identify them further. So our actual project going forward is we're taking a look at people that have got different levels of LPA, because the people with high levels of LP(a) are particularly prone to vulnerable plaques. So having established this method, the next steps are for us to compare how people's plaques look when they've got the sort of vulnerability indices of the LP(a). We've also been quite able to do this as comparative biology as well, which has been quite exciting and has actually exceeded what I thought we would possibly able to do. This is our human plaque here. We find very similar sort of signatures. This is, for example, a cholesterol ester in the rabbit, which is more of an early stage model of atherosclerosis. But just in very recently, Claire McLeod, one of our PhD students, tried this in a mouse model of atherosclerosis, which I have to say I didn't think was going to work because I thought it was going to be too small. But what you can see, here's your H&E with a little bit of plaque around the edge, and actually we are able to pick up, this was actually a steroid we were looking at, but there are a number of lipids that look like that as well. So we haven't fully interrogated this yet, but it is possible to do this in very small animal models as well, which is quite exciting. So that's a work in paper in progress that hopefully will be completed soon. So I think I hope I've excited you by mass spec imaging and shown you how tricky it is to get from fluid biochemistry to tissue biochemistry, but it does open a lot of doors and understanding. And I just need to thank all the team, I think particularly Shazia, Sva, for the imaging, Diego, who worked with me previously, and I showed you work of Don Livingston, Howland 2 and Rita Upretti today about the 5-alpha reductases mainly. And thank you for listening.
Thank you, Ruth. That was a fascinating tour de force of the complexity also of these lipids. I mean, really tiny changes in structure that have absolutely massive effect on their biological activity.
Yeah.
Really impressive, isn't it? So a question from Bill.
Okay, Ruth, very, very, very nice talk. So I was wondering if you saw any ketosterols in the plaques.
I think I'm going to have to come back to you on that one, Bill. So we haven't done that derivatized. So we haven't looked at derivatized. We've only looked at the mouse derivatized plaques. We haven't analyzed the human and the rabbit for steroids. We probably need to do that by the GRT approach. So the answer is I can't tell you about the humans. The only one we've done derivatized is the mouse. So I could take a look though. I could take a look at the mouse data set.
Those papers by Brown and Jessup, they talk about the 7-keto in the plaques. So when you were looking at the 5-alpha dihydroxytestosterone, do you have interference there with the second isotope of testosterone?
Yes, that's yes. So we've spent ages getting the chromatography right there so that particularly with the derivative causing the two the two peaks. So we've spent a lot of time with the careful chromatography to try and sort that out. So yes, you do. And that, so you have to be really careful with your chromatography doing that. Yeah. Okay.
What about ion mobility in front of the mass spec after the after the moldy? What do you think?
Yeah. So we've actually analysed the plaques that way, and we thought we were going to see a lot of our peaks split in two. So actually they didn't. We actually found that most of our peaks didn't split. So I don't think we were doing it wrongly because we're relatively new to the technique. So I think it did give us reassurance on our molecules that we were able to be more confident that we had one molecule there. But actually most of what we gained, we already had we felt the fragmentation was pushing us in the same direction. So it gave us a second bit of corroboration. So we have used it, but it hasn't yet separated out anything that's helped us understand something differently, I would say. So we don't use it routinely.
Okay,
So we go back in. So what we do is we make a list of molecules and we think that could be more than one. We'll go in and look at it really specifically, and we'll probably also try to buy the standards in that situation as well. So we're not using it as a routine screen, we're using it as a check.
Okay, thanks, Ruth. Lovely talk.
Okay, Rashmi, you can ask your question. It's just a comment, actually. Excellent presentation, very good research work. Sorry, I should have read that before I unmuted.
Thank you.
Okay, I've got a couple of going right back to the start of your talk. I was really interested in, now I may have got this the wrong way around, but from what you were saying, with obesity, cortisol levels are higher?
In tissues, but not in the circulation. So
yeah.
what you actually
Why?
So well, I touched on the enzyme. So that enzyme 11-beta-HSD1 is upregulated in adipose, and it's also which is one of the major contributors there. Actually, in the liver, it isn't higher, but it's higher in adipose, and then adipose is quite a strong contributor to
But it's the enzyme that removes cortisol, isn't it? So
no, there's two of them. The one that generates cortisol is higher in the adipose tissue.
So I suppose I was thinking it was the pathway that removes it, which was
So there's two of them. Yeah,
there's two, right? So then I guess the thing is, there's a kind of a chicken and egg situation, is there, a little bit, because as cortisol goes up, appetite may increase. So which comes first, the obesity or the higher cortisol, and what's the interplay between one causing the other, and then the other way around?
I know. I think we've been involved with some sort of genetic studies trying to unpick this type of thing and looking at the glucocorticoid receptor sensitivity. I don't think you can unpick it, but we have a colleague at the moment who's trying to do this in large animal models, where you can get a little bit more of a control and actually sort of control the levels of glucocorticoids and see what the outcomes are. So you can't do that in humans. So I don't know, Val. I think it's a chicken egg, but there's no doubt about it. If you interfere with it, you suppress and correct some of the
and also like the underlying causes of how those enzymes are affected by obesity. So certainly 5-alpha reductase is under insulin regulation. You see that very clearly, and some of the glucocorticoid receptors, some of the cytokines are definitely affecting its expression. 11-beta-HSD1 as well, insulin. I think growth hormone has some roles there as well. So there's been quite a lot of work done looking at the regulatory elements, but it comes down to the insulin axis, and certainly 5-alpha reductase, that's been the major controller we found was insulin. And I mean, like, how with obesity, higher levels of stored fat being metabolically active, contributing to all of this, is that driving a lot of this somehow?
So I think you get a lot of steroids. You get a lot of steroids dissolved in fat as well, so they become quite a major sump of glucocorticoids. So actually, when people are adipose, you've got the weight of the adipose, not just the enzymes producing it. Actually, they pick it up into the lipid droplets. So we visualized that with cars, that you can see the steroids going into the lipid droplets. So the turnover actually in adipose tissue, glucocorticoids stay in there for days when they go in. So it takes a long time to get adipose to steady state with tracers, and then it's like 3 or 4 days before you actually get turnover. So adipose doesn't quickly respond in stress, but it holds a pool of glucocorticoids that's really quite substantial.
Okay. And one last question. We have a question, but I'll ask my last one, which was around, all of these. Obviously, I'm more familiar with sterol esters, cholesterol esters. Do they exist for a lot of these other lipids?
Esters?
Fatty acyl esters, for example.
I don't think so. You get the glucuronides and the sulfates. You get the standard conjugates. Can't think of it. Fatty acid esters, actually. No, I'm not going to say absolutely not, Val, but it's not something I'm.
Either they don't exist or no one's looked for them.
I wouldn't like to be dogmatic, but not that I'm aware of.
Interesting. Okay. All right. So we have a question from Gerard. Okay. What are the storage conditions of the sample? Are they fixed or not?
So for the small molecules, it's frozen. We take them as quickly as possible onto liquid nitrogen and cryopreserve them like that. So that's to stop the small molecule moving. Yeah, so that's really important. But for larger molecules, you can use fixed sections. It's the structural components. You can actually use fixed ones. It's just if we put our tissues through fixative procedures, you'll wash all the steroids out. But what you can do after multi-imaging is go back and do histology staining. It's just got holes in it, that's all. But you can quite largely see the structures there with the standard histology stains after. Okay, it's just not quite as good as formalin bedding detail, but we find it's pretty good actually.
Answer your question, Gerard.
Yeah, so it's fine. So my question was, when you use, for example, you cannot use ethanols or methanols or something like that. But I think that you can use maybe formaldehyde because when you use a certain type of fixation, it's possible to use oil redo staining procedure. So you can localize the, if you use staining procedure, which can be used to visualize lipids, especially oil redo. That means that you can use the methods that you develop by mass spectrometry. Yes.
So you mean on the same section, Gerard? Is that what you mean?
Yes,
so,
yes.
So yes, I don't know if we've actually done oil redo afterwards, but we've done various different stains after the mass spec imaging. And what we also sometimes do is take the adjacent section, and you can do it that way as well and just superimpose, as long as they don't change in size by the way that you're processing them. So yeah, I haven't tried oil redo after the imaging. I'm not quite sure how that looks, but maybe.
Because if it is possible to have a simultaneous image, not a simultaneous, but a contiguous section, that means that you could, maybe it could be possible to have a view of the lipid distribution in the tissue section, and after, with the methods developed, to have another view of the distribution of sterol in the section. But another question, what is the resolution of in terms of microscopy, comparatively to microscopy? What is the resolution? That means that you can see, that you detect, in fact, the profile, because you use a laser. It's a technical point. That means that you have a nebulization of a part of the tissue, and is it a big part of the tissue, or what is the volume, for example, the minimum volume that you can detect, and consequently to have an idea of the part of tissue which is taken in consideration to do a measurement, because so what is in microscopy, we speak about voxel, that means X, Y, and Z. And with this technique, what is the size of the volume where you do the measurements?
So you're right. It depends on the laser steps. So how much you move the laser and the nature of the laser. So the one we use, we can move it in 50 micron steps. Once we start moving less than that, we start to overlap. Now, you can oversample if you want, but you're overlapping. Now, that is not the limit. There are other ones that are common at 15, and there are other sampling methods called like SIMS that go down to nanometers. So it depends on the technology you've got. But if you go really small, you get less sample, so you only really measure the really abundant things. So there's a balancing act, and SIMS doesn't lend itself to all types of molecules either.
Okay, thank you.
Okay, thank you very much for your question, Gerard. So I think we've just come up to just over the hour, Ruth. So it just remains for me to say thank you very much for an excellent webinar. It was great to host you, and we'll be back next month for the next webinar. So thanks, everybody. Bye now.
Bye.